It's really a pleasure to be here, to have an opportunity to meet people in person. I think is very special, but particularly it's special as a physician because this is kind of why we do what we do, which is the opportunity to help people. One of the things I want to tell you up front is that I've never done the specific myeloma crowd talk before. I tried to ask Greg earlier about what was the audience like and what should I talk about, and he said give it a talk to as if you were giving it to a remedial first-year med student. The problem, Greg, is that there's no such thing as a remedial first-year med student. It's so hard to get into medical school these days. But what I'm going to try and do is actually give you an overview of, or set the stage for my colleagues who are going to follow in this. And here's what I'm going to do is essentially give you a quick basic refresher about the immune system. This is a field that I've worked on for 25 plus years. And then just leave you with 5 top messages. I can't remember more than 5 things at once. So I'm just going to give you only 5 messages from this talk. So I think we all know about the immune system, particularly since the pandemic, as being a very important component of what protects us against pathogens. Just some basic concepts. Immune system is not one thing, but it consists of many different kinds of cells, at least 30 to 40 different kinds of cells. And we broadly characterize them into what we call the innate immune system and adaptive immune system. And you'll hear more about these kinds of things from further talks. In the adaptive immune system, which is the one that's specialized because it mediates specific recognition of the target, also has the capacity for immunologic memory, which means that when you actually were exposed to something, the immune system will remember 20, 30, 40, 50 years later about that exposure and try to protect you from that. And that I think also relevant for the perspective of cancer is the concept that the immune system actually has a capacity to evolve and adapt. Why is that important? Because we also realize that our enemy, which is the cancer, also has the capacity to evolve and adapt. And so you need the solution to have at least all the features of what the opponent can throw at us, but be one step ahead. And that's why I think the immune system perhaps becomes an important component of the fight against myeloma. The second sort of broad overview I'd like to give you is this concept of the so-called cancer immunity cycle. And this is a concept that has evolved over now 20, 30 plus years. But we now appreciate that essentially all cancers are recognized by the immune system. They all have antigens because they have abnormalities in their genome, and that creates abnormal proteins that can be recognized by the immune system. These cancer cells, if they are killed by chemotherapy, the radiation therapy or surgery, for example, that creates the opportunity for recognition by the immune system. These dying cells then are recognized by a set of cells called dendritic cells, which are specialized for presenting and processing and presenting those antigens. And that goes to a part of your body called lymph nodes, where these antigen-presenting cells meet another kind of cell, the T cell I told you about with the adaptive immune system. And that sort of, if you will, tells the T cell or instructs the T cell that such an antigen is something that we need to make an immune response to. Those T cells will then, after they are activated, will then go back to the tumor and eventually get into the tumor and kill the tumor cells. And essentially, what has been appreciated is that there are several aspects of this so-called cancer immunity cycle where there are breaks at the present time. So there are systems wherein the immune system is suppressed by what's called checkpoints such as PD-1 and PD-L1 in the cancer milieu. And there have been studies, particularly in solid tumors, where just simply taking away those breaks has led to remarkable and durable responses in many patients with cancer in the last decade. There are studies where we've tried to enhance recognition of cancer cells by T cells. And you'll hear more about that later as well, with what we call CARs or bispecifics. And you will hear how these therapies are beginning to change how we treat several other hematologic malignancies, including multiple myeloma. But what this tells you also is the concept that we're only at the beginning of this revolution, because we're only, for example, tackling one or two aspects of this cycle. To be able to optimize this, and those of you who are involved in manufacturing a product, something, you need to make sure that from the first to the last step, everything works to make a product. And so in this case, we have plenty of opportunities to improve the process through attacking it at multiple fronts and making it harder for the immune system. So just keep those basic concepts in mind to get a context of where we are today and where we want to be perhaps a decade or so from now. So as I said, I'm going to actually limit my talk to just 5 insights. The first is something that actually, an observation that we had made almost now 20 plus years ago, when we found that even advanced relapsed refractory, so-called high-risk myeloma, was sensitive to killing by the immune system. This was, in fact, at the time, difficult to convince our community of, because at the time it was thought that perhaps cancer cells are too close to normal cells. It was really hard for the immune system to figure out what's normal, what's abnormal. And it's really, immune system is really meant for pathogens, viruses, bacteria, things like that. But I think it's become very clear now that it's actually very hard to find a cancer cell that cannot be killed by any immune cell. If a cancer cell becomes resistant to an immune cell A, it may probably be more sensitive, in fact, to immune cell B. And so I think this concept tells us that even in advanced disease, we will have the opportunity to engage immune system, whether it's our own immune system or somebody else's immune system, to be able to target the cancer cells. And that has the potential to achieve durable responses and even cures by appropriately targeting the immune system in that setting. The second concept that I think we, again, have sort of studied now over the past 10 years, and I think I personally believe that this is really where we will make the biggest progress in the fight against myeloma, is not to actually cure myeloma, but to eradicate myeloma, which is that it doesn't exist. And the reason why we can do that is because we know that all of myeloma originates from a precursor stage. So even before we diagnose any patient with myeloma, that patient has had abnormal cells in their body for at least 10 years on an average. And that condition, as you know, is called MGUS. What we do now know is that these earliest stages of MGUS are in fact recognized by the immune system much before any pathologist or anyone else can diagnose or find those cells. So the immune system is already be able to detect these very early immune cells. The problem is that then we have a 10-year period over which this stuff is evolving. What we have learned from infecting mice with what we call chronic infections, and humans with chronic infections like HIV, for example, that immune system keeps reacting to a chronic stimulus But after a while, the T cells in particular enter a phase that we call, for lack of a better term, exhaustion. So they don't work as effectively as they used to when they were earlier on in the stage of the game. And to be able to maintain that response for, in the case of mice, for example, for many months, or in the case of humans, for many years, you need a capacity of those T cells to have what we call stem-like features to be able to maintain those chronic immune responses. And what we are finding now is that as we track people with MGUS and with myeloma, that the people with MGUS already have features of exhaustion, and that as the disease turns into myeloma, the stem-like cells, in fact, are getting depleted over time. So essentially, imagine a situation where you have an army of people, and you already made them run half a marathon, and now you want them to compete in a battle. It would be better if you didn't make them run half a marathon before you took them to battle. And that's kind of what we do currently with the immune system when we're trying to tackle cancer to some degree. And for that reason, I feel personally that the future for us, perhaps 20 years from now, will be in trying to prevent MGUS and its evolution to myeloma. And largely because we're trying to get a head start on that whole question. The third, I guess, insight or lesson is something that anybody who's interested in real estate knows very, very well. It's all about location, 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 right? So why do I say that? First, because we know that myeloma is multiple. Why do we call it multiple? We call it multiple because it likes to grow in clusters. Myeloma cells don't like to live alone. They like to grow as a group of cells together. It is because of that reason that they actually are able to form things like bone lesions. They're able to form, we can actually see that in MRI scans and PET scans where you have what we call hotspots of things. What we're beginning to appreciate now is that this biology or the capacity or the need for myeloma to grow as clusters creates a very distinct spatial interaction with the immune system. So when the tumor cells start to grow as clusters together, some of this data is actually sort of not even fully published yet, so we're hoping to get this out into major scientific journals in the coming months. But the concept is that when the tumor cells grow together, they actually don't let these T cells get in. The only way the T cells get in is if they have a visa or a passport to get in. So they need to be able to recognize the target. And as I said, there are only some T cells that recognize specific targets. So the way in which we can overcome that is to give a whole bunch of T cells a visa or a passport. You can do that by creating artificial recognition systems such as CAR T cells or bispecific antibodies, where you actually have one arm of an antibody recognizing the target. But what that means is that the target is going to be the key in terms of getting the cells into the tumor. And that really makes the case helps to explain why bispecifics and CAR Ts, at least in the first generation, are actually beginning to work in myeloma. Now, obviously, it also tells us, this work is also beginning to tell us that it is also dependent on or also modified by what we call this immune cell state. So it's the same kind of concept that if you have a very exhausted cell, which has already run half a marathon, then it's not going to be as effective, it's not going to last as long. And this is where we believe that the issue of persistence will be solved. which is that you get a response to therapy, but you actually can't keep the T cells in there for too long. And eventually the cancer comes back. And the way to figure that out is to figure out ways in which we can actually keep those cells in that microenvironment for a longer period of time, or give them the right amount of appropriate stimulus, if you will, and keep the bad cells away to be able to maintain that response. And this has become the #1 question in our field, because as you will see, we get a very high, quote unquote, response rate to CAR Ts and bispecifics, but we do not get an equally high cure rate as of 2021. And the solution is to actually find a way in which we can keep the cells in there for longer periods of time so we can get more durable responses. The fourth sort of insight I'd like to mention is that it is none of this is going to be a one-size-fits-all. And we already know that from many things that we do in medicine, but we specifically know that for the immune system. The way your immune system responds to something depends on what's called HLA molecules. And all of us, except for our twins, which most of us don't have, actually have different HLA molecules. So right off the back, everybody is different. We also know that by when we study the immune system, we cannot find two people with the exact same immune microenvironment in their cancer. We also know from studies that I'm sure you've heard about that no two myelomas are the same. Each myeloma has distinct pattern of mutations, et cetera, et cetera. But what will make a difference in terms of the future of immunotherapy, and you'll hear about that, partly from Yvonne and others, is that it will create the opportunity for differences in expression of the CAR T targets. So the most common CAR T target that we currently utilize is against BCMA. It's a B-cell maturation antigen, but there are going to be at least 3 or 4 more different kinds of BCMA, of CAR T targets that are going to be in clinical trials in the near future, or they're already in clinical trials. But there also are other mutations that these tumor cells can exhibit, and some of those mutations are potentially targetable by the immune system. All of these, of course, will then impact how we specifically approach a engineered immune system, if you will, for that patient. The states of the immune cells, the nature of immunosuppressive cells, even the microbiome, which we think now have enough evidence to suggest that it actually does modify the properties of the immune system in general, as well as prior therapies that the patient has received. All of these will impact immunotherapy in the future. Why do I say that and why do I know that? The reason why I say that and know that is because it affects the response to all your common vaccinations that you get every day, not all the time. And we do know that all these things impact how you respond to vaccines. And so it will likely also impact how you respond to other immunotherapies in the future. And I think as a physician and as a scientist, I think I personally believe, and this is for which I don't have any data, because all the other 4 things I told you actually had real data behind them, that in my view, cure and eradication of the cancer will require the immune system. I say that because of the reasons I already pointed out earlier, which is that it is the only system that has long-term capacity for maintaining an immune response. And so you don't have to keep taking a medication all the time. And it can keep adapting to if the cancer decides to adapt. But also perhaps more on a personal level, I think that we shouldn't be doing research if we are not trying to go for the cure. And so, from that perspective itself, I think it's really important that we think about the cure. And in my personal opinion, The only way I can see that as of right now is a strategy that we actually engage the immune system, largely because we do not have a strategy to surgically resect multiple myeloma, as it happens in other solid tumors. So that's all I'm going to say. Just go back to the 5 top insights. I think the immune system provides an hope for long-term control. It provides the hope for prevention. I think it's already impacting therapy. Some of the therapies that we currently use, the so-called IMiDs, thalidomide, Revlimid, pomalidomide, and also bortezomib, Kyprolis. All of these drugs we've shown in the past engage the immune system, and that the cure and prevention will require the immune system. I'd like to thank the many colleagues that help us with our work, both at our institution and other institutions, and the NCI, LLS, and the Riney Foundation for our support. Thank you so much.